All right, well, welcome everyone to our EvoFunPath seminar, and we are very happy today to welcome Matt Fisher. So I'm going to tell you a little bit about Matt. First of all, the first time I met Matt, I think it was at that meeting on Roscoff in Bretagne, one of those emerging fungal disease meetings. It was a beautiful setting. We were by the ocean, and then we met again in Ascona in Switzerland a few years later. And I hope that we're going to get some more of those meetings in the near future. Matt works on emerging pathogenic fungi, and he heads a research group at the Department of Infectious Disease Epidemiology at the Imperial College in London. And Matt was actually a natural historian. He grew up in Zambia, and he says that as a barefooted child, he was naturally attracted to natural history. And then he went on to get bachelor and PhDs in Edinburgh, but in zoology. And actually, I didn't know that or never looked it up. But then Matt saw the light when he went to Berkeley to work with the mycologist John Taylor, and then he metamorphosed into a fungal biologist right there. And it's a trajectory that he continues now at Imperial College in London. Matt has already some Canadian connections, so he's part of this CFAR program on fungal kingdom threats and opportunities. He's also an honorary fellow of the Institute of Zoology and received also the Berkeley Award of the British Mycological Society. But I'm guessing that's not the same Berkeley where you did your postdoc, something completely different, right? Matt is a highly cited researcher. He's wrote some very impactful papers that I keep citing, such as the emerging fungal threats in nature, and then more recently his worldwide emergence of resistance to antifungal drugs in science. So Matt, it is a real pleasure that you accepted to give us a seminar today, and the floor is all yours, and I hope that you can share your presentation with us.
Fantastic. Thanks, Richard. It's great to be here. Lovely to be back in Canada. I spend a lot of time in Canada these days because the CFAR network is quite an unusual network internationally in that it's so incredibly generous, and it's a very good process of bringing international minds into the Canadian research setting, and also leveraging small pots of money that we can all use around the world to answer the questions that we feel are important. So it's been a very transformational experience for me. I'm giving Canada the thumbs up. Let's have a look. Okay, so you can hopefully see my first slide coming up now. Does that look okay, Richard?
Yes, good to go.
Great. So I'm here at Imperial College London in the UK. We've just built some new labs, and looking out of my window here, I can see the arch over Wembley football stadium from the 11th floor. So we're very lucky to have these new labs. We've moved from St. Mary's, where I was the first mycologist to work there since Alexander Fleming, back in the 1940s, who made a serendipitous discovery which kickstarted the antimicrobial revolution. So really amazing to work in St. Mary's, but those buildings are past their retirement date and have been sold, and we've been moved on. So I'm part of a Centre for Global Infectious Disease Analysis, which is mainly targeted at the heavy-hitting viral diseases that you'd expect, including that one called COVID. We've been central to the global analysis of that threat through the modeling expertise of Neil Ferguson and people like that. But my speciality is the Cinderella of emerging infections, Kingdom Fungi, which is extraordinary, and I'm preaching to the converted here, very interesting arena with a diverse set of emerging pathogens, of which I'm going to focus on amphibian chytridiomycosis here. From all the stories that I could talk about, I think this is the one that encapsulates the power of genomics to really unpick a very cryptic epidemiology. So let's just go back in time to when I was a child. So this is my entry into the field of mycology. Me in the left in 1976, and I'm holding Termitomyces giganticus. This is a termite mushroom. It's farmed by fungi, and it produces these immense fruiting caps. This is the biggest mushroom in the world, and that one actually was the biggest mushroom in the world back when we bought it off a Zambian lady by the side of the road. We entered it into the Guinness Book of Records and didn't actually detail it well enough to get it in, but this species is the largest mushroom, and they're delicious to eat. So skipping through many years, I eventually ended up in Berkeley after my PhD in parasitic nematode population genetics. And this is when John Taylor and Tom White took me under their wing and said, you know, Matt, great to work on, you know, parasites of guts, but fungi are a lot better. And they really opened my eyes to the use of the nucleic acid approaches which I developed in Berkeley, but could then apply to the fungal kingdom. Now, Tom White was actually Kary Mullis's boss when Kary Mullis envisaged and developed PCR. And because Tom White was interning in John Taylor's lab, he was able to say, look, John, there's this amazing technique where you can amplify DNA and then you can sequence it using the Sanger reaction, and you can actually start to do phylogenetics of your fungi. And so those early papers were really key in population genetics, and they really started in Kingdom Fungus because Tom was in John's lab. Anyway, so I pitched up years after that, and but the lab was firing along on all cylinders, and I fell into this into the world of fungi from the point of view of developing polymorphic markers with which to dissect their population genetic structure and apply that to the epidemiology of fungi, fungal pathogens. Now, while I was working on valley fever, Coccidioides immitis and Coccidioides posadacei, that's a species split that I completed through the use of nucleic acid markers, there was emerging on the scene, we're talking the late 1990s here, that there were widespread diseases in the environment, fungal diseases, that were proving very aggressive in wildlife populations, specifically frogs, and bat white nose syndrome wasn't on the scene then. But I was in the lab when David Wake, who sadly died last year, he was in the Museum of Vertebrate Biology, came and started talking to John Taylor about this, what turned out to be a fungus that was wiping out amphibian populations across the world. And John was very enthusiastic and said, yes, of course, we need to work on this. And me, being the postdoc in the room, got to pick up those research threads, which I didn't really do a very good job of for a while until I got back to Europe. And then I started isolating the chytrids and working on their population genetic structure. So this is an article that, you know, this is a training network that you've got going here, and there's of course a lot of there's a whole deep tool bag that you can dip into to work on the genetic structure of whatever fungal pathogen you're interested in. So this is something that I wrote with Rhys Farrer a few years ago, but it just kind of details the generalized workflow that you would apply to a new emerging fungus that you don't really understand very well. So EFP here at the top is an emerging fungal pathogen, and you have a species hypothesis. So at that point, there will either be a reference genome, it's been previously sequenced, and you have a pretty good idea of what it is, but it's emerging for some ecological reason. But because there's a reference genome, then you can use those tools that you can work with, basically applied to that reference. So you can align to the reference, you can call variants, you can go straight into phylogenomics, population genomics, GWAS, QTL mapping. So that's the whole pipeline on the left-hand side. However, if there's no reference genome, then you've got to generate that whole genome yourself, and then you're into the de novo genome pipeline. You've got to RNA sequence to give you your open reading frames, and then you've got the genome annotation, and eventually you're moving that pathway from the right onto the left. Now that's actually changing quite fast, because the ability to work on pan-genomes, to actually take reads and assemble them, even short Illumina reads, and assemble them into a reference genome for all the variants that you're calling, and to essentially do a population genetic analysis without having a reference genome is now become possible. And this is proving really interesting, and is something that you need to think about actually how you can go straight into the de novo genome construction and pangenome analysis to do the population genetic analysis without having to rely on these references, which may be fantastic, but at the same time, they don't capture all the diversity of the species that you're working on. So yes, don't forget about pangenome analyses. So I'll take you through my kind of line of inquiry on the amphibian parasitic chytrids, because I think it really will show you at least how this particular research line evolved as we acquired more and more genetic insights into what was an entirely unknown organism to begin with. So the backdrop to all of this is that amphibians have become increasingly rare across the 20th century. And it was a key conference in Canterbury in the late 1990s, where herpetologists put all their stories together and recognized that there were these enigmatic declines occurring in pristine environments where there was none of the recognized threats such as deforestation or ecotoxicology. There was some unknown process, and this kind of clicked on through time. And to cut a very long story short, eventually the etiological agent was discovered. It was one of the basal chytrid fungi in the Rhizopediales. So this is is it 9 phyla now in Kingdom Fungi, with the Chytridiomycota there. They've been around for over 500 million years. They were on the planet before amphibians dragged their way out of the seas and became the first land-dwelling vertebrates. They're flagellate organisms, and they cause this very aggressive disease in amphibians, amphibian chytridiomycosis. So these flagellate zoospores essentially swim through aquatic media, and they can detect and burrow into and reproduce within frog skin, and they can go on and infect the next amphibian. And this is a very famous plot from Karen Lipps's PNAS paper detailing her transects through time in El Cope, Panama, just showing what happens to the biodiversity of a neotropic rainforest when BD hits. And you can see this biodiversity dropping off the edge of the cliff. And it's completely tragic in these neotropic rainforests these days, because where there used to be swarms of amphibians, they're now almost entirely silent. So there was the discovery of BD, and then much more recently, the sister conspecific taxa Batrachochytrium salamandrivorans appeared on the scene. This was specifically identified in fire salamanders in the Netherlands and then Belgium. And again, it was an incredibly aggressive infection. You can see here this key study showing the decline across a few months of fire salamander population in these in the Dutch forests. This is the cluster of infections. It's now sadly increasing spreading very rapidly across those northern European forests. And there have been little sparks found down into Spain. So there are some long distance dispersal mechanisms going on there. It will probably spread across all of Europe, and the lethality in these fire salamanders is incredibly high. Where did it come from? Well, it looks most likely that it came from Vietnam. It's been found there in low prevalence infection in the salamanders there without causing any disease. So we know quite a lot about the kind of the origins of B cell, but BD remains much more cryptic. And of course, this is the much larger story. The B cell story is a very specific case of a single introduction into a European ecosystem. But BD is right the way across the planet. And this is a really good paper by Ben Shealy that I was lucky enough to be involved in, which essentially is it's just an accounting of the species and timescale that BD has exerted across the amphibian populations on planet Earth. And BD has caused a decline of many hundreds of species and the complete extinction of at least 90, although there's uncounted others there. And this emergence across the amphibian species of the world, I mean, as far as we can tell, it started in the early to mid 1970s. So we're talking sometime between the start and the middle of the 20th century, and peaked in the 1980s, although there are still species which are declining as a result of the spread of BD. So from its original description, this is the time scale of discovery of BD and B cell. The World Congress was in 1989, and that's when the threatening process was identified. Joyce Longcore, who won the American Microbiology Golden Goose Award for the discovery and naming of the pathogen, then moved into more mechanistic studies of where the chytrid was and what its impact was, and whether there were action plans could be exerted to try and mitigate its spread and threat, and then more work on the origins, which I'll describe here, and impact. So there's now a really hefty literature underpinning this whole kind of timeline of research from the initial papers in the late 1990s. So it really is. It really has gone from zero to hero in terms of understanding and of this, which was once a very unknown phyla of basal fungi. So here's the first lesson. Fungi are worse invaders than even cats, rats, and other infectious diseases. They have the demonstrated potential to burn down enormous amounts of the tree of life. So you have to take fungal pathogens very seriously. Now, the epidemiology of BD and chytridiomycosis is about as complicated as it could possibly be, because absolutely everything matters. And I think this is a general principle that you can apply to most fungal pathogens is, the normal epidemiologist in the department that I'm working here of infectious disease epidemiology, they're dealing with a human and a virus such as COVID-19, and it's actually really quite predictable. You can put together mathematical models which will predict what's going to happen with a fair degree of accuracy and what mitigation of methods are going to work. And this is essentially the process that we've been engaged in over the last 2 years. Well, you can't do anything like that for fungal infections generally, certainly not the BD, because there's everything dealing with a cold-blooded host, so abiotic factor is incredibly important. The actual ecology of the amphibians, the communities that they're embedded in, the alternative hosts that the chytrid can transiently infect and amplify in, the microbiome of the organism itself. Remember, the host is always a holobiont of some sort. And then the actual genetics of the chytrid itself all play a role. So good luck with getting your head around any of this. And this is certainly what I've spent the last almost 20 years doing now for this particular epidemiological scenario, because you can dissect all of this, but to actually really understand what's going on, you have to integrate across all of these factors. And that's what makes this such an incredibly fascinating picture. Anyway, but for genetics, you have to get your organism, and that's not so easy, is it? Because when you're dealing with a chytrid that lives in the skin of frogs, then you've got to somehow culture it, and you've got to go and catch the amphibians. So key to a lot of what we've been doing is actually developing fairly simple methods of going into an environment, taking a biopsy from an animal, and then catching this not only quite rare, but also difficult to culture organism. So that little video there described one field trip to French Guiana that France was kind enough to fund. We managed to get 2 isolates out of that trip from the 1,000 or so amphibians that we caught in those wonderful, those are the most isolated rainforests on the planet. But that was good, because what could have been a disaster of a trip was successful, because we got what we came for. Other parts of the world aren't so easy to find nice French helicopters. So this is a field trip to Madagascar, where I've met the worst roads on the planet. And that's as far as our Land Cruiser got before we had to start unpacking our expedition and putting it onto the heads of local villagers who were kind enough to take our ewers of liquid nitrogen up the mountains to where we were able to find the frogs that we came to study. So this is just going to say that when you see a genome in the NCBI database, just be aware of how much effort actually went into putting it there. We never really know the backstory of those sequences that we find, and it's always very interesting, and it's often very hard-won data. So, but the important point here is that if you're able to work within a community framework, if you're able to develop a tool, a technique that you can train others in, and you can help them get it working, then you give some love, you receive some love, and this is what's happened with this amphibian work, is that there's been a real buy-in from the global community of herpetologists and researchers, and that has enabled these early genomic databases to be developed, which have answered a global process. With any research group working in isolation is not going to get the answer; they'll only see a little bit of it. So you have to think and work collaboratively if you're going to attack the big problems. And so that's a really important point that I'd make to young researchers is of course you're focused on your own particular piece of the story, but the more that you're able to reach out and to work with that expertise across the world, the bigger your vision is going to eventually be. So by leveraging that very kind of simple protocol eventually this phylogeny came together, which was the work of people like Rhys Farrar and Simon O'Hanlon as the main data analysts of this global data set. And it was really fascinating because our naive expectation was everything was going to be reasonably similar because this infection had clearly blasted across the planet, but in relatively recent time. But that turned out to not be the case. There was some really interesting lineage diversity there. For instance, this thing here called BD Cape, which is what we now know to be a highly Africanized lineage, although it's had some sparks into other parts of the world. That's the island of Majorca, where it was introduced there into midwife toads. And it's even been found in Central America more recently. Here we have this very interesting lineage, Asia to Brazil, which is found both in Brazil and Asia. So there's clearly some expansion event there. We have here BD Asia 1, which I'm going to talk about in more depth later. But this is the one that really matters, BD GPL, the global panzootic lineage. And this is what's caused most of the trouble across the world, because it's the weed that's infected all these environments that we've been working on. It's also, generally speaking, a more highly virulent lineage than the other ones. And it's associated with all those key signature declines and extinction events. That's not to say that it's the only one that can cause disease in the wild, because we are working on some interesting epizootics with BD Cape, but really it's BD GPL which has caused the problem. So with these genomes, we've answered some kind of key questions which have been kicking around, which is first of all, when did BD GPL emerge? And to answer this sort of question, well, you can use it using Ben Shealy's type of data set. You can just go back in time and ask people when amphibians became rare, and do essentially a meta-analysis of decline data. It's a bit unsatisfactory, isn't it? You'd like to be able to twin that with another method, and that's what you get with the genomes. And simply speaking here, if you've got isolates which are separated in time, then you can calculate the number of mutations which have accrued since the split, and you can calculate a time to most recent common ancestor from that. That's exactly what we did with BD, but it's not as nice, simply evolving virus. So all those wonderful methods which amazingly intelligent people such as Andy Rambow and have developed for use within the beast paradigm, they don't work in these scruffy and highly complex eukaryote genomes which characterize our fungi. So we put some effort into twinning our nuclear reads with a mitochondrial genome, which we had to create ourselves from the reads that we were assembling from our chytrid isolates. We used PacBio for this, and it's a really extraordinary mitochondria, isn't it? It's linear, so nothing in fungi is ever simple. It's enormous, so 300 kilobases, and it has some unitigs as well. So it's really quite a wacky mitochondria. Anyway, we're able to assemble that and annotate it. So what that nice big mitochondrial genome gave us was a phylogeny, mitochondrial phylogeny, which is independent of the nuclear phylogeny. You can see there the different lineages being mirrored in that mitochondrial phylogeny, and look how tight BD GPL is there, which I've circled in red. It really hasn't accumulated a lot of diversity over the time that of its expansion, but it has accrued some, and so there's a significant R squared there. So we have an increase in diversity through time. So you can calculate a clock rate on that. In certain parts of the nuclear genome, we've had we've had some loss of heterozygosity in what's a haploidy genome down to a haploid. And so you can clip out those regions of the nuclear genome and get a fairly clear clock signal out of that. So what we're able to do here is put together those two different clock rates to get a composite date of emergence of GPL. And it turns out to be some time in the first half of the 20th century. So those are the two distributions, one for the supercontig in red and the mitochondrial DNA. They're not perfectly aligned, but they're not bad, are they? And then that's Ben Shealy's estimate for the when the declines really started to crank in the 1950 onwards. So it does seem to be that BD GPL expanded across the planet in the first half of the 20th century. So this is an Anthropocene time point. This is when we really started globalizing the world and spreading objects around, including amphibians, and this appears to be the main seeding event for the most destructive lineage. We're also working on time dating the other lineages, but that's a little bit more. That also requires and we're making headway there too. Okay, so we've got we've got this the nucleic acid estimate for when BDGPL emerged. Then question then becomes, from where did this lineage emerge from? And there's been all sorts of candidates bandied around over time. North America, Europe, South America, Africa looked very promising from some early identifications of BD from South Africa. What does the genomics say? So Tajima's D is a nice metric here. It identifies populations that are evolving neutrally and haven't been through big demographic processes. And using Tajima's D is often a good way of spotting an ancestral population that's in some form of equilibrium. It's probably recombining, and it hasn't gone through any dramatic bottlenecks. So these are the expectations for Tajima's D. The answer that we got from our analysis of all our genomes was that BD is an out of Asia event. So here's Tajima's D for the Asia 1 lineage, and you can see that it's flat as a pancake, sits basically right on 0, whereas the rest of the lineages, Asia 2, Brazil, Cape, GPL1 and GPL2, there's a couple of clades within GPL, are all over the spot. They're all over the place, and this shows the dramatic series of demographic events, spread events, the sparks that have moved genotypes around the planet from the ancestral population. You can also look at this through the eyes of rooting the lineages. If you root against BD lineages against BSAL, then these Asia lineages fall out as basal, so that puts them in an ancestral position. The Asian lineages are also incredibly diverse, and this is, to me, one of the most telling pieces of evidence that you've actually hit on the ancestral diversity. We didn't actually get very many isolates of Asia 1 in the end, but those that we did get, 7 in the end, were 4 times more diverse than any other lineage, which was sampled, many, up to 100 isolates. Those two, in GPL, you'd only find 1,000 SNPs. So great diversity, and then that diversity that you find in Asia 1 is present in the other lineages. So this is fairly good evidence that Asia 1, that GPL is essentially a spark out of Asia, and it has been plucked from this deep well of diversity that is in the continent. Where in Asia? Well, we don't know. The isolates that we got were mainly collected from South Korea. So at the moment, we're suggesting that it's from around this region, but obviously we have to sample much more widely across Asia. And that's very difficult to do because the amphibians don't die there, because they've been infected by BD for many millions of years. both B cell and BD, so they don't die. They're highly tolerant or even resistant to these pathogens. So you've got to search very hard to get these Asian lineages of chytrid. So that's my second lesson, is that the genomics gives us the hotspots of fungal diversity that exist as pathogen pumps into the rest of the planet. And how do they pump these genotypes out? Well, the genomics gives us the answer for that as well. What we're doing here is essentially identifying pairs of isolates which are very highly related, and those are linked by some form of transmission event. We can also go into trade, and we've had students over the years that have been working in the Heathrow Animal Reception Centre and going around pet shops, and they're doing this in America and Europe, and collecting the BD isolates and sequencing them. So those 4 pictures at the top of the slide are all species of commercially traded amphibians where we've isolated BD GPL from. So this lineage is widely embedded in the global amphibian trade. So you don't have to hunt hard for a mechanism of spread. It's right there in front of your eyes. But it's not just GPL, is it? So these next 4 species were also, their chytrid was isolated from trade, and they were BD Cape. So we've got another lineage which is being widely occurring in the trade. And we have a very well-known spark event from South Africa up onto the island of Majorca. That was a captive breeding program gone horribly wrong. And then we even have recovered BD, both lineages of both Asian lineages from traded amphibians. So this is quite a remarkable finding, isn't it? That the known phylogeographic diversity of amphibian infecting BD, all of it can be recovered from traded animals. So we're really very good at spreading this organism around. Our biosecurity isn't doing very well. So essentially, what you're looking at here is a model of an explosion of diversity out of Asia, sometime in the 20th century, genotypes being moved around the planet. The fungus hasn't lost its ability to recombine, because we're spotting hybrids of the different lineages in the environments that we're working in. So those 3 red stars at the bottom are isolates of BD, which are hybrids between GPL and Cape. We find them in South Africa. So that's where these sparks out of Asia are recontacting and recombining, and you can see that they're creating new combinations of alleles. So you're generating diversity as you go along. You're essentially recapitulating your ancestral diversity plus those mutations which have been picked up through time. So this data, while scientifically interesting, has also been really important in making the case that you have to treat the amphibian trade seriously. Biosecurity matters, and this has now resulted in some trade bans for specifically salamanders in Canada, in the United States, and now Europe. So focusing on salamanders is because this is the main vector for B-cell, and B-cell has a very limited distribution to Asia and those northern European forests, and isn't something that we want moving into new continents, especially those of North America, where you have incredibly high biomass and biodiversity of plethodontid salamanders. So, if you're going to hang on to that biodiversity, you need to keep B-cell out. And the genomic data has been incredibly important to making that argument. So trade matters. We live in a highly globalized world, and it's incredibly bio-insecure. This, of course, has profound consequences for health, both wildlife and also plants as well. So I'll just close by talking about some of the comparative genomics that we've been doing within the species, because I've been talking up until now about population genetic analysis of BD that we've been able to do from those global genome collections. But of course, you can start to compare the two pathogens themselves and see if there's any interesting genomic features which explain their pathogenic lifestyle. So here we go, we've got BD and B cell, and we've got two environmental related rhizopedialia chytrids, Homolaphyctis polyrhiza, HP, and Spizellomyces punctatus. So what we were able to do was to infect this peculiar animal that's a crocodile newt, Tylototriton. We use this species because it gets equally infected by both BD and B cells. So you can do really nice expression analyses in it. And then of course you can compare the two genomes of B cell and BD. B cell is a genome that really likes to secrete. It's got a lot of genes with secretion profiles, about twice the number of BD, which has got more than the environmental chytrids. And another striking feature of the two pathogen genomes is they're absolutely full of repeats. So there's a high diversity of repeats across all of the chytrids, but more in B cell and BD. But on the right-hand side there, you can see the percent of the genome which is picked up repeats, and it's way, way higher, strikingly higher for the two pathogens. So protein degradation is a key virulence factor for the chytrids. They love to eat their host. So you can see in this picture of a fire salamander here, B cell essentially ulcerating, rotting down the skin of the fire salamander. And that's because they've taken a very easy trick. So these are chytrids that like to feed on protein. They want more, they're endlessly hungry for it. And so they've taken the easy genomic route to just expanding their protease genes into gene families. And so you can see here in green the BD M36 proteases, but look what B cell has done. It's just blown them up, and this really explains why B cell is such an effective protein degrader. This is a key virulence factor for both chytrids, but most so for B cell. So B cell is loaded with secreted genes, a lot of which are these the M36 proteases, but there are many more. Are these virulence factors? Well, this is where we all share a problem in the fungi that we work. Is it's easy to identify open reading frames, and the putative proteins that they produce, but actually then to figure out what they do is really hard work. And that's where we've stopped with these. But what about those repeats? So this is a really great paper that came out of Sophie and Kamoon's lab. And and this is where they really set into place the idea that genomes didn't have equal evolutionary rates across them, that they were compartmentalized. And this is often called the two-speed genome model. And what it essentially says is that the repeat-rich and gene-sparse regions of the genome are evolutionary accelerators. It's where the most of the adaptive action occurs. And certainly when you have a pathogen which is coupled to a host and diversity matters, then you find that the virulence effectors concentrate in these rapidly evolving regions of the genome, essentially driven by the repeats themselves and their rapid transposon type modes of adaptation. So do we see those are in Phytophthora genomes? What about these repeats in the B cell genome? And this is where Rees and Teresa in Exeter have done some really good work with nanopore by building a best-in-class genome of B cell and looking at where those repeats, those huge number of repeats that we find co-locate with the known virulence factors, the amplified M36 gene family. So what these plots are showing is that as the distance between genes increases, that they're filled in with repeats, then you start to find a higher association with genes that we expect to be associated with virulence, in this case M36s, but also those other unknown virulence factors that we're spotting in these genomes. So there is evidence for dynamic evolutionary compartments in B-cell, and this is the first evidence for two-speed genomes in an animal fungal pathogen. But it does raise the question of whether or not these evolutionary compartments in genomes are a more general feature of eukaryote genomes, and that wherever you have strong adaptation, then you have this compartmentalization into strong selection, and adaptation then becomes coupled to an evolutionary motor, such as the expansion of various repeat families, transposon related. So here's the take-home message 4. These gene family irradiations are a cheap and cheerful way of driving pathogenicity in BD and B-cell, and they've taken a very obvious evolutionary route there. But there are differences in the epidemiology of the organisms which can probably be associated with these with different effector repertoires that we still have to understand more about, and that there is evidence for two-speed genomes in these fungal pathogens. So that's a rip-roaring trip through the world of chytrid genomes. I think what's fascinating to me about chytrids is they exemplify much of what make fungal pathogens incredibly interesting and also incredibly perilous organisms, just because they are so creative in exploring the evolutionary ecological niches that we're offering them to in the Anthropocene world that we have created. And this is my final message. It's not mine. It's from Dr. Ian Malcolm in Jurassic Park, where he says immortally, "John, the kind of control you're attempting simply it's not possible. There's one thing the history of evolution has taught us, is that life will not be contained. Life breaks free, expands to new territories, and crashes through barriers, painfully, maybe even dangerously." But well, there it is. And I couldn't really have said it better myself. That's exactly what we've seen with chytrids. We have to really be aware of fungal threats, and it'd be really nice to get ahead of the curve, wouldn't it, to try and anticipate and protect what biodiversity we have left. And certainly some of those trade bans for North America are playing well to that particular tune. So here is some just some kind of the bodies of literature which have been put together, which I think are quite useful when taking a walk through the great fungal stories that are out there. The one on the left is from a Royal Society meeting that we wrote back in 2016. So it's starting to show its age now, but I still really love the series of publications we put together for that very important meet. And then this colloquium report from the ASM is also really valuable, and it takes a lot of the thinking that we're using in the CFAR program and putting it into this colloquium report in a highly readable manner. And it would just be before me to just thank at least some of the collaborators which have been key to working on these chytrid genomes and the chytrid genome story. But of course, there are so many more that don't make it onto this slide. And of course, we've also got funding as well over the years. So thank you very much. I will now unshare my slide, and I'm very happy to take any questions.
Thank you very much, Matt. That was fascinating as usual. So we, bravo, bravo.